In the previous module, I gave you an example how PCR technology has had an effect in the clinical side and improving human health. Now I will give you an example of how PCR technology has helped the society, human society. I'm sure you have already heard about DNA fingerprinting technique. I'm going to tell you how this is done. You are probably aware it because it has been in news that a small sample of a victim or a person can be used to verify the authenticity of that person. So let's look at this. Now, let's say that we use 2 primers in a reaction mix, and when we run the gel, we get several bands. Here I'll draw these bands. We used 1 primer, and this is our DNA lane. This is the gel, and we got several bands when we ran that PCR reaction. Only 2 primers. It should have only produced 1 fragment. This implies that the primer is binding at sites. Our primer is specific. There's no problem there. The target DNA is of variable length, and it has constant regions to which these primers are binding. It so happens that there's some sequences in human beings which are variable number tandem repeats. What are those? Let's look at the tandem repeat part first. These repeats, they're specific sequences. I'm going to just draw one sequence. Whatever the sequence is, we'll talk about that later. It is repeated in tandem. Tandem, as I mentioned before, it is sequentially one after another, like cars in a train. These are tandem repeats. They're constant sequences. Let's make a sequence here. AT, ATT. This is just an arbitrary sequence. This sequence is repeated 3 times in the diagram I have made. These sequences are repeated variable times in variable numbers at different locations. Now please keep in mind that this sequence is also present here. So if we make a primer which is complementary to this sequence, on the other strand of the DNA, it would be, of course, TA. So if we make these 2 primers which are complementary to these 2 regions, they will amplify this segment of DNA. Now imagine if the tandem repeats they're repeats, but they are variable number. In this case, there were only 3. In other cases, we may have, say, for example, 20 tandem repeats, one after another. In other instance, we have the tandem repeats repeated 30 times. In other instance, we have tandem repeats repeated 70 times, so on and so forth. When we run the PCR reaction, these repeats, because these repeats have the constant sequence, they will hybridize the primer and produce bands which are variable in length. And when we run them on the gel, we have said the DNA moves according to the size. So the size of these DNA fragments will be different. Now let's look at the genetics of it. We inherit variable number tandem repeats just like other genes and alleles we inherit from our parents. We have 2 chromosomes, one from mother and other from father. So 50% of our chromosomes are from mother and 50% are from the father. Here is a case where we want to determine paternity of a child. We are not sure whether this child is the real son of a particular person. So what we do, we can run these VNTR primers. We'll take this DNA sample from father, the mother, and the child, and we will run them. We will run a PCR reaction with the VNTR primers. When we run that, we will generate these bands. It so happens that the banding pattern is unique to every person. The probability 2 people will have the same pattern of banding, VNTR fragment bands, is 1 in a billion. So we take the mother's DNA sample, we run this procedure, we get the VNTR bands, we repeat the procedure with the child, and the probable parents, the probable fathers of this particular child. And now we see if this child is actually son or daughter of male 1 or male 2, it will also have some of the bands from that male. In our case, let's see, this is a child. He has inherited this band, for example, from his mother, and this band has been inherited from this person. This male number 2 does not have this band. I will erase this so that you can see this again. Now you can see this band. This child has inherited this band from the mother. This band has not been inherited from mother because in mother lane you don't see that band. This band has to come from father. And this male has also the same band, but male number 2 does not have this band. If we look at these bands sequentially, another case would be this band. The mother does not have this band, but this male has the band. Male number 1 has that band. Male number 2 does not have that band. So if we run this DNA fingerprinting for paternity, and we can determine paternity of a child, because a child will inherit 50% of VNTR bands from one parent and 50% of the VNTR bands from the other parent. So these VNTR bands are inherited in Mendelian fashion. Another use of this technology, as I mentioned, is crime scene investigation. I have said the probability of 2 people having the same VNTR bands is 1 in a billion. So say, for example, if there was a crime and there was a victim, and victim had some sample of the aggressor or the criminal who committed that act, it could be some simple things. Saliva. If the victim scratched the aggressor, the skin under the nails of the victim. If there was a struggle, victim may have pulled the hair of the aggressor. Any sample that may have DNA in it, we can take that sample. Now remember, we have the power of PCR technology. Even if it was just one cell, one nucleus, we can amplify it millions of times and generate VNTR fragments. Now the rest of the job is left to the police. They can determine who would have the probable cause. So they round up the suspects and we run the VNTR fragments, VNTR PCR on them, and we generate these banding pattern. So you can see it is easy to see here that the blood stain matched with this person. So this person is the most likely the criminal who committed this act. I will show you an actual image. So here is a crime scene investigation. Victim, suspect number 1, suspect number 2. See the DNA sample at crime scene. We have to see if it matches the victim or not. It did not match the victim. It meant that it belonged to the person who committed the crime. And the suspects were rounded up. And suspect number 1's bands are identical to the bands which were obtained from the sample of from the crime scene. So this is another way PCR has had impacted our society.